Hi again, everyone. Welcome to Part B of Chapter 10 on photosynthesis, where we're going to be following up with some of what we talked about last time when it comes to electromagnetic spectrum and the chlorophyll that's involved in the absorption of that energy. We left off with a simple experiment where if we split white light, presumably white light in this case, we're going to get an even range of different frequencies, ranging from a very long wavelength in red to a relatively short wavelength in purple. And if we open up just the slit for green, what we'll see is that because chlorophyll does not absorb green, nor typically will reflect it or not absorb it at all, when we shine it on a photoelectric tube, we get a high current. So near 100% of the energy that came in here is now coming through and striking this tube. On the other hand, if we move it to the blue spectrum, what we see there is that virtually all of the energy is absorbed by the chlorophyll rather than allowing it to transmit through, and so the energy levels are relatively low. When it comes to chlorophyll, we actually have two different kinds of spectra, and I'm not talking about the red and the blue here in this. We have what's called an absorption spectrum, that is to say, which wavelengths of visible light will the chlorophyll most absorb. And we can see here for chlorophyll A, which is in red, that the best wavelength is somewhere in probably around the 430, 435 range, and then somewhere in the red range, somewhere around 650 or 660, 665. Those two peaks represent the greatest amount of energy absorbed if we were to shine that wavelength of light. And so in that situation, if you had a plant with chlorophyll A, you'd be best to have some sort of a bulb that would shine in a violet blue spectrum and or a little bit of red. On the other hand, we have something called an action spectrum, which shows the relative effectiveness of different wavelengths, and it parallels closely this absorption spectrum, but an action spectrum reflects a specific type of experiment. Why do we have an absorption spectrum versus an action one? That's because leaves and organisms that undergo photosynthesis have multiple pigments, okay? Have multiple pigments that are all capable of absorbing energy. So what we see is we see the different absorption spectrum of chlorophyll A, chlorophyll B, and then the the orange color in carrots. You see the word carrot here. That's where we get this. Beta carotene is a member of the carotenoid pigment family. Also has an absorption spectrum, and so these guys sum up. They add together, and what we see is that the rate of photosynthesis is generally higher in this where we see all of these peaks, generally lower where we see all the troughs. And you can see there's a little more shoulder here. Why? Because maybe the carotenoids are contributing to photosynthesis right around here. And the same story over here. We have a couple of peaks, and so our relative ability to undergo photosynthesis is higher in this range. Action spectrum is derived from the original experiment. That is to say, Engelmann had these aerobic bacteria. These are bacteria that can only grow in the presence of oxygen. And he also had algae. And so since photosynthesis makes oxygen, he grew these things together. And what he found was at certain wavelengths of light, he got a lot of aerobic bacteria growing. And at other wavelengths of light, he got hardly any bacteria growing. This was in anaerobic conditions, and the only oxygen that was available would be from photosynthesis. So the action spectrum comes from an experiment like the Engelmann experiment, which you'll read about in your book, where we had a combination of this algae and aerobic bacteria, and it would only grow when the photosynthesis was occurring and oxygen was released. A description of Theodor Engelmann's experiment, and you should be able to explain what was going on in this experiment. He had filamentous algae, which is a photosynthetic algae. He also had aerobic bacteria. And what he found was that the aerobic bacteria only grew when you had photosynthesis occurring, which would produce oxygen. So he started out with very low oxygen concentrations, and then as photosynthesis occurred, the oxygen concentrations would rise, and the aerobic bacteria would grow. We have that experiment with the aerobic bacteria, but what would happen, and this is a really nice thought experiment to help you make sure you understand the concept of not only photosynthesis, but the idea of facultative or obligate aerobic and anaerobic organisms. Let's say instead of using aerobic bacteria, we used anaerobic bacteria that could only grow in anaerobic conditions of which oxygen would be poisonous. So what would we expect to see? We wouldn't see this where when we have lots of oxygen, we'd see lots of growth. We'd actually see the opposite. We would actually see a peak somewhere out here and a trough somewhere around here. We'd probably see something along like this, and then maybe a little rise at the end. Something not unlike this rather poor representation. I'd have it rise a little bit there at the end. Okay, but that's kind of what an action spectrum would look like using an obligate anaerobic bacteria in the presence of our filamentous algae. That's a great thought experiment. If you really understand something, you can come up with theoretical experiments to test your hypothesis out. Now, chlorophyll A is the predominant chlorophyll. So we have that in the dark green spectrum here. That's one is the most prevalent in most leaves and plants, is chlorophyll A. Chlorophyll B, on the other hand, broadens the spectrum. So if we find ourselves in competition with other plants, chlorophyll F is sort of the same way. If we find ourselves in competition with other plants and in very, very competitive photosynthetic situations, using other pigments will help broaden the spectrum so that we're not just relegated to very, very specific wavelengths. We can actually work in different wavelengths that we normally wouldn't be able to by developing those other pigments. Carotenoids, they also will absorb. Mostly we think they are for protective reasons, but that doesn't mean they don't contribute to photosynthesis, because they do oscillate and capture energy and transfer it to a reaction center. It's just that carotenoids are much less efficient at that, and mostly what they're there to do is to help protect what we would call photobleaching. Photobleaching is a common phenomenon in microscopy, especially fluorescence microscopy, where the light is damaging the pigments and causes them to bleach out. And chlorophyll is no exception to that. So what we have is we have these carotenoids to help absorb some of that extra energy. Maybe some of it gets used for photosynthesis, but a lot of it just gets dissipated as heat. We can really see this in the fall colors. You can see the different pigments showing up as the chlorophyll A and chlorophyll B get degraded first. The other carotenoids and other compounds, of which, like for instance, flavonoids, we might hear about that in our diet. They can be quite a good nutritional supplement. Carotenoids we find in carrots. We can also find them in things like sweet potatoes and other types of vegetable products. Anthocyanin and other types of redder carotenoids give us that deep red color that we see like in maple trees. We've already talked about this, but the reaction area of chlorophyll is up here at the top in this porphyrin ring, suspending that magnesium ion. And then we have this hydrocarbon tail that helps hold it in place in the thylakoid membrane. Now, normally chlorophyll is in those reaction centers inside a leaf. And what happens is as it gets excited by light, it transfers that energy into the reaction center, that special chlorophyll molecule, and then those electrons are handed off into electron transport. But if you isolate chlorophyll, what you will find is that it will go from a ground state when it's in the dark to an excited state, and this is unstable. So if you extract chlorophyll from the leafy environment, the cellular environment, that energy actually has to go somewhere. And so that energy goes away by virtue of what we call fluorescence. Fluorescence is a way of bleeding off energy, just like bleeding off heat. You shine a light on something, you'll feel it warm up, and it's radiating heat as it's giving off that energy. But it's not only just heat. Heat's a form of electromagnetic radiation. We can also give off forms of light that we call fluorescence. So if we isolate chlorophyll, we shine a certain wavelength of light on it. We can not only get heat from that solution will not only warm up, but it'll also glow in a very, very specific wavelength. Just like a bell rings at a specific tone, you know, in the example that I used in class, a bell will ring and it has a given tone. We will also give off a given frequency of light. So let's look at this. We're going to excite that chlorophyll, but you have excited electrons. If it can't give those electrons away, it has to release that energy in some other form, usually heat and light. And that's what we have shown here. This is actually chlorophyll, which we're used to seeing as green, right? Well, we're shining UV light on this chlorophyll, and it's actually glowing red. It's giving off photons of light at a red wavelength and is probably warming up pretty well as well. Putting this back together, we have those chlorophyll A molecules, we have some chlorophyll Bs and carotenes and carotenoids and so forth, all vibrating and getting excited in the presence of photons of light. Handing off those excited electrons rather than releasing that energy directly, they're handing off those electrons to this reaction center, this P680. That P680 becomes a will hand off those electrons to an acceptor and then obtain electrons from water in order to replenish those lost electrons. That's what makes up a light harvesting complex, a combination of chlorophylls and carotenoids and so forth, a reaction center, all waiting for those photons of light to show up where they begin oscillating. The electrons begin oscillating. They have a lot of energy in them. We hand them off. It hands off to an electron acceptor, and then we'll resupply those electrons from water, that electron donor. So that primary electron acceptor is a molecule that allows, in the case of Photosystem II, for those electrons to enter electron transport. In the case of Photosystem I, however, our reaction center is a little different, and our electron acceptors are a little different as well, because we're handing it off to NAD, first of what we call a ferredoxin, and then an NADP reductase enzyme. So we're using the sun's energy to excite those electrons, we're handing them off, and then we're harvesting the energy in a couple of different ways. We've seen this picture a number of times already, and that illustrates the importance of it and that you need to work on this. This requires some work to get this and to get all of the pieces of the puzzle together. So the photon of light excites that light harvesting complex. It'll hand off some excited electrons to that reaction center, that special pair of chlorophyll A's, which will then hand off those electrons to this reaction center complex. That's what enters electron transport if we're talking about Photosystem II. Then what it's going to do, it's going to rip off electrons from that water. And here's just a picture. You can see how complicated it gets. And you can see those chlorophylls embedded in there. It's a solar panel, is really what it is. It's an array of light-sensitive molecules that begin oscillating in their electrons when photons of light show up. We have talked about this verbally, but I wanted to put it down on paper that there are two types of photosystems in the thylakoid in most plants. We have Photosystem II, which is the first step. This is the one that's going to create oxygen. We're going to rob electrons from water after we've given up electrons to the reaction center for electron transport. So the photon of light causes electrons to get excited. We hand them off, and that we harvest the energy. Once we hand this off, this P680 plus is very, very strong oxidizer and will rip those electrons right off of a water. That causes oxygen to want to react with itself, creating diatomic oxygen. And we're going to have a few pairs of protons, which we're going to use to pump or create a proton motive force. So photosystem II, and it's called photosystem II because it was discovered second, has that P680, has best at absorbing at 680 nanometers. So that's why we call it P680. So once it's been excited and hands off those electrons, it then becomes P680 plus, and it's P680 plus that's going to steal those electrons from water. And after electron transport, which is not shown here, that electron enters the photosystem I, which has a special chlorophyll center that is most excitable at 700 nanometers, a slightly different wavelength of light. And again, we get those photons, we excite electrons, only this time we're handing off the electrons to this FD, which means ferredoxin. Ferredoxin, F-E-R-R-O-D-O-X-I-N, ferredoxin. And ferredoxin in turn will hand off the electrons to an enzyme called NADP plus reductase. NADP plus reductase. It is reducing NADP plus. It adds those electrons and the protons come along for the ride. We get NADPH. So P700 is what we call this one, but it's almost identical. The main difference is the wavelength of light and the electron and where the electrons are handed off to. To this point, we've shown you the basic process of electron flow. That is to say, electrons from water through the photosystem II, electron transport where we're pumping protons, photosystem I where we're handed off to NADPH to go to the Calvin cycle. This is known collectively as linear electron flow. It's not doubling back on itself. It is not cycling. It is, albeit a crooked line in this picture, but it is a line that is linear in nature. So we have a kind of a Z thing kind of going on. And we're making ATP and we are reducing NADP plus. Those are the products of what we call linear electron flow, which is your normal photosynthetic electron flow. However, plants, while they don't have brain cells that we know of, are not stupid biochemically. If all we had was linear electron flow, this plant would be very, very vulnerable to water deficit, exceptionally. Because what would happen if we didn't have water? We wouldn't be able to do stuff. We would shut down and die. And several billion years of evolution has made plants and all organisms exceptionally well adapted to their environments, which can include water deficit situations or situations that we need to do something different in order to meet metabolic need. However, there are times when a plant will need to undergo what we call cyclic electron flow. So if I show you this picture right now, we're going to go into this in a little more detail. But instead of handing off the electrons to NADP plus reductase, we cycle those electrons back around through electron transport. But we'll get to that a little bit later. That's the cyclic. If linear would mean we go here and then hand it off to NADP reductase, we're actually handing it back over into the electron transport chain. But we'll get there a little bit later. Right now, what I want to do is review what we've covered to this point. So the next several slides are about reviewing all of the steps of photosynthesis. So we have our photosystem II. We have our light harvesting complexes here, different pigments. They oscillate and vibrate and their electrons get excited. And what happens is that P680 will then hand off electrons to a primary acceptor, becoming P680 plus. When it's P680 plus, it's a very, very strong oxidizer. So strong that it will steal electrons from water, creating diatomic oxygen, the molecule of oxygen, create a few protons there while we're at it, which will be used for chemiosmosis. Meanwhile, those electrons get handed off. Those electrons get handed off to the electron transport chain. And so here we are, we're handing it off to the electron transport chain. I think this is a phytochrome, but I'm not sure, I forget now. And it's a cytochrome complex, just a lot like electron transport in mitochondria. And we're pumping protons. We pump those protons, we'll use chemiosmosis and ATP synthase to create ATP. And so it's a proton gradient across the thylakoid membrane as opposed to the inner mitochondrial membrane. And that diffusion, that movement, that proton motive force is turning that turbine of ATP synthase, forcing inorganic phosphate onto ADP. So that's what's going on here in steps 4 and 5. Once those electrons have been handed off, they are now the donor electrons for photosystem II. And you can see that photosystem II is a little offset. You see that photosystem II is a little bit higher in the vertical plane than photosystem I. That's because those electrons never quite are as low. This is a higher energy set point. They're actually using the y-axis as energy level. So we're losing energy. We'll harvest ATP from that energy through chemiosmosis and proton pumping. Then those electrons become the donor electrons for P700. And again, as light bounces through the light harvesting complexes, those electrons will get excited. We'll hand it off to an acceptor. When that acceptor gives those or takes those electrons, P700 will accept more electrons from the electron transport chain. So each electron is falling down an electron transport chain from that electron acceptor up here at the beginning of electron transport. So it's a kind of a little bit of a busy figure, but we're exciting P680 and we hand off those electrons into electron transport. And this is where we're going to pump some protons. Okay. Then we're going to excite those electrons again momentarily in the photosystem I, where we then hand off those electrons. Instead of to electron transport, we're going to hand it off to ferredoxin and this NADP plus reductase, which will transfer those electrons onto NADP plus, creating NADPH. So we're making NADPH, which is an electron carrier, and that will take it to the Calvin cycle down the line. If you break up this figure into its individual pieces, each little piece is, shall we say, edible, rather than trying to eat the whole pizza at once. Start over here at 1, explain to yourself what's going on here. Then what's happening when we get from 1 to 2, and then what's happening at 3 and 4 and 5. Remember, this is not direct, this is indirect. We're doing chemiosmosis here. Then what's happening at 6 and 7 and 8. I would never ask you to draw a figure like this, but I would ask you to tell me what's going on at each of these individual steps. Describe to me in your own words what's happening. So here at the end of 6, what we're doing is we're handing off electrons to ferredoxin in 7. It's then being handed off to NADP plus reductase in 8 to reduce NADP into NADPH. Doesn't require super terms, but having just the right number of correct terms will make it much easier for you. So work on the terms, work on the process. I admit that you may not remember this for the rest of your life, especially if you're not involved in plant biology or that sort of a thing. But this is the kind of process that we go through with certain types of therapies and certain types of problem solving in healthcare. So being able to think sequentially and think dynamically is a very, very important skill, and one that I think needs more emphasis. That's one of the major reasons why we would teach this dynamic process to you, even though I know that most of you don't really have a ton of interest in plants. And that's a shame, because I think plants will ultimately save us. If we understand plants better, we can take care of them better, and then they will take care of us better. The figure in the previous slide is too complicated. Start here conceptually. Start here conceptually. Get these concepts down, and then you can go to the other figure and break out the pieces. So this is a nice conceptual understanding, but we do need to have a little bit of specifics to get that practice of learning how to learn specificity, because specificity is everything in therapeutics. If you give the wrong therapy to somebody with a condition, you may not it may not only not help them, but it actually may make matters worse. So learn the concepts and then apply them to the specific terminology, and you'll get a long, long way in almost any class that involves medicine or therapeutics or so forth. And here we have the systems in the actual membrane. You can see that water originates inside the thylakoid space, and that we're actually building up protons a couple of different ways. First of all, we're building up protons from the photosystem, as P680 plus is stealing electrons from the water. It's creating protons. We don't even have to pump them. When we do electron transport, we're pumping some protons there, so we're getting protons from Photosystem II, and we're getting some protons from electron transport. That gradient builds up. We have that proton motive force, and we're going to make ATP from it. But those electrons from the electron transport are going to enter Photosystem II and get excited, hand it off to ferredoxin and NADP plus reductase to make NADPH from the oxidized NADP plus. And both the ATP and the NADPH are going to be a part of the Calvin cycle, which occurs in the stroma outside the thylakoid, but still inside the chloroplast. It's going to be in the stroma of that chloroplast. So that concludes part B of chapter 10. Next time we're going to start talking about how we take that NADPH, we take some of that ATP, and we use these cyclic reactions known as the Calvin cycle in order to make this G3P sugar that then becomes glucose down the line. So we'll start there next time. And so thank you for your time. As always, if you have questions or are having difficulty envisioning this stuff, let me know, and I will. We can arrange a time to help you better understand that. There's no reason you can't get this. It just takes the right kind of process to make this stuff stick. So again, thank you, and I'll talk to you soon.